So now that we've looked a little bit at chemical properties, reactions, and how they operate, let's kind of take it a step further. So now we're going to talk about endergonic and exergonic reactions and some of the theories of how those might operate in the cell. Well, actually do operate in the cell. All right, here we go. So transformations of energy are how cells are able to perform work. And when we're talking about work, you got to think about cells as being, you know, a little bit like a factory. They're building stuff, they're breaking stuff down, they're doing all kinds of, quite frankly, work. Some of those things are going to be maintenance, maybe making new cell parts, maybe breaking down old cell parts. Also, a really important one is replicating cells. So that's going to mean we're going to be forming a lot of chemical reactions. We're going to look a little bit at how those operate. Let's start with the two types of energy. The first two types of energy changes that are important to understand. So the first are kinetic energy and potential energy. So kinetic energy is the energy of being in motion, actions happening, right? So if you were to have a ball on the side of a cliff, the kinetic energy would be the ball falling off of the cliff. However, potential energy is a little bit different. It's stored energy. It has the potential to do something. It's just not doing it right now, right? So potential energy would be having a ball on the edge of a cliff and it almost tipping over, but not. It's holding still. It has the potential to do something, but it's just not doing it right now. Right, so two of the other forms of energy that we talk about a little bit in biology, and sometimes a lot in biology, is thermal energy, which is a form of kinetic energy. We measure it as heat, right? So you would take a patient's temperature, for example, and that tells you a little bit about the chemical changes or the work that's being done inside of a cell or body. And then the other one that we talk about in biology, we're not going to discuss it so much in this particular class, is photosynthesis, okay? And this is when we're harnessing the power of light, right, in order to fix carbon and make sugars. So for chemical energy, this is a type of potential energy. And so think about this as the way we store energy in our food, right? Sometimes we're storing it in fatty acids. So fat cells have tons and tons of this. Sometimes it's stored in carbohydrates like sugar. But what it is it's energy that we're saving for later, right? So whenever we have a set of carbons, right, hydrocarbons actually, these little bonds right here, we're going to wait. We don't need it yet, but we're going to hold on to it, right? So it has the potential to become a bond that's broken up and the energy is released, but we don't need it, so we're going to save it, okay? So it's potential energy. All right, and one of the most important ones that we'll talk about during the semester is going to be the idea of ATP, which is an energy transfer molecule. It's really efficient. It's kind of like the spending money of the cell. So among some of the definitions that we'll go over when we're talking about the idea of energy transfer is going to be that of thermodynamics. So basically, this is whenever we're looking at any kind of group of matter, collection of matter. In our case, a lot of times it happens to be like, you know, a cell, maybe, or a group of cells, for example. And we're going to look at the energy transformations that are occurring maybe within the cell, or that group of cell, or that ecosystem, whatever it is, whatever that collection is. Okay, so it could be how they're using energy, maybe they're storing energy, maybe they're growing rapidly. And so all of these different changes are going to have repercussions in terms of what kind of potential energy and kinetic energy is being transferred, right? And so there's two kind of words that you also need to know. So a system would be the matter under study, like the cell or the individual or the collection of cells or the ecosystem. And then the surroundings would be like all the things that are around it, right? So when we look at it, that's kind of the dynamic part of thermodynamics, right? So dynamic, so there's multiple interactions, right? So at least more than one system that's being studied, and because of that, we have the specific system, in this case our cell, and then the secondary system, which is considered any of the surroundings. So you're going to need to know a couple other concepts too. So some of these are some laws. So you need to know the first law of thermodynamics. So the first law of thermodynamics says that all the energy in the entire universe is constant. And basically for you, what that means is energy can't be created or destroyed. You have to get it from somewhere if you need it to power your cells, for example, right? So that's how we think about it in a biological context. So we have to pull energy from bonds that are already in the environment or from bonds that we can make from components of the environment. And that's how we can then rearrange them, save them for later, or open them up and create kinetic energy or use kinetic energy. All right. The second thing you need to know is the second law of thermodynamics. And the second law of thermodynamics deals with the idea of entropy. Okay? And so when we think about the second law of thermodynamics, I want you to think about this. Every time you, say, eat a Snickers bar and you digest it, you're tearing apart bonds. You're increasing the disorder of the universe. We quantify that at least using heat, right? We just talked about that. So heat is one of the ways that we quantify energy changes in living systems. So that's why when you go to the doctor, we take your temperature, right? So that's why it's like almost always a monitored variable in most biological experiments, okay? So it gives us an idea of how much work or entropy, right, is being done. So the work would be from breaking chemical bonds, right, releasing potential energy, transferring it into kinetic energy, so we can do work, doing things like, I don't know, making replacement cells, making replacement cell parks, maybe just getting energy to do more activities. And so when we break those bonds, again, that's how we release or increase the disorder or the randomness in the universe. So we're dealing with chemical reactions at this point, right? And so we looked at kinetic energy, and we also looked at potential energy. And now here's some related concepts. And those are the idea of exergonic and endergonic reactions, right? So for exergonic reactions, I want you to think of like X, right? So like exergonic, right? Exit, right? So these are types of reactions that we're breaking bonds open, and so that energy is exiting, okay? So for endergonic, right, like in, I want you to think of like going in, like going into, right? So we're putting energy into bonds, okay? We're saving it for later. Usually this is how we're making things like potential energy. So taking a closer look at exergonic reactions. Exergonic reactions, again, remember, oops, there it is. Release energy, right? So we're breaking bonds. That's what's important. So they're releasing the energy in covalent bonds. Covalent bonds are the highest energy types of bonds. They store the best, right? And so it takes a lot of energy to actually break them open and to harvest that energy for other uses. So some ways that we talk about it, at least, is going to be burning wood is going to be releasing the energy because you're breaking the bonds between different types of carbohydrate molecules, and we're getting heat, and we're also getting light, which are some other ideas that we just talked about, other ways we can quantify these energy changes. But then when we talk also about like, cell maintenance, which we do a lot of in this class, think about cellular respiration, right? So this is taking out energy from food. So when you're eating that Snickers, you're going through a whole lot of steps, which we'll go through later on. So Krebs cycle, electron transport chain kind of stuff. It's going to take a lot of steps to break open these different bonds. And we're going to kind of produce another molecule that sort of saves the energy, but really just moves it around to where it needs to use. So we save it in ATP. Okay, that's our energy transfer molecule. That's how we take energy out of, say, a glucose molecule, and we're able to store it right here in the triple phosphate bond and shuttle it around to whatever part of the cell needs to use it. Maybe the cell membrane needs to be repaired. Maybe we need to make some more DNA, so we can take it to where it needs to go, so it can do the work that needs to be done. It's really nice. It's specific. The other thing that's nice about it is you can reload it and use it later. It's a great molecule. We'll learn more about it later. So here's this canonical graph that you're going to see pretty much everywhere when we deal with things like exergonic reactions. And sorry also for the formatting here. For some reason, all programs still don't want to talk to each other very friendly. But let me just explain to you what's going on anyway. What this side is talking about is how much potential energy is stored within a molecule, right? Or anything, really. In our case, I use the Snickers a lot because I'm always hungry for them. I know it's bad, but I am. Anyway, so there's a whole lot of potential energy stored in, say, a candy bar, right? Lots of fat, lots of sugar, all the bad/good stuff. And so if we have our total max calories right here, and we eat the Snickers bar, what happens is if we need to use that energy right away, so we need to power our lungs and our hearts and our brains, and we're going to go run a couple miles, all that, what's going to happen is your body's going to say, "Apes, look at the energy there. I'm going to take it out." And so what it does is it starts digesting it and sending it through cellular respiration and breaking down those larger molecules, those storage molecules, so it can release as much of that energy as it can for the work that needs to be done in your cells, just to maintain yourself, just to get all that running done, right? However, what's left are products. So after we've stripped out a ton of this energy, what happens is we're still left with our same set of atoms and some of these newly transformed molecules. Okay, but these now have less bonds between them, and that's an important point. The lower the amount of bonds are, then the lower the potential energy is of the products. And so that's how we can find how much energy is released. So that's just the idea. But inversely, let's take a look at something else. Now this is an endergonic reaction. We talked about this a little bit ago. Remember, we're going to put energy in, right, to an endergonic reaction, right? So that's kind of, I know the spelling's totally off, but you get what I'm saying. So what we're going to do is we're going to make bonds. We're going to store energy for later. That's how we're going to do it. All right? So we're going to have to put energy in, input of energy. Remember what happens when we store energy in bonds. We're not using it. Using it would be kinetic, but we're doing potential energy. So we built bonds. We've added bonds into molecules, okay? We made new, larger molecules. So these endergonic reactions usually start out with kind of small potential energy, but as we are able to add onto that molecule using covalent bonds, we're able to increase the chemical energy that's stored in them, or the potential energy that's there, just in case we need it later. So you've seen this, right? Except it's backwards. So this is the exact opposite of an exergonic reaction. That's it. They're just inverse. So in this case, we're taking smaller reactants. So this might be an example where you have, say you ate the Snickers, right? And you had X amount of sugar in that Snickers. But instead of going for a run and cleaning your house and doing all this stuff, you do what I love to do, and let's take a nap or a few naps. And so what ends up happening is, since you don't need as much energy, you're able to kind of build bonds, right? So you're adding bonds in, and so you're changing that sugar now to fat. So fat is a really high energy storage molecule, because instead of just having a couple of carbons, right? So a couple of carbons is not, well, you get the idea, right? A couple of carbons linked together, you now have many carbons linked together. So that's many more bonds. So it's a lot more energy. And that's why it's so hard to kind of do things like burn off all of the calories that you consumed if you haven't done something like go out for that run or exercise, right? I know, sad stuff. But this is an endergonic reaction. We've put energy into our molecules, making them larger and giving them more potential energy to use later, hopefully use later. So now that we've learned a little bit about the binary ways that we can transfer energy and describe them a little bit, let's talk about some broader concepts of the cell. So the thing is every minute, every day, your organism, yes, you, and other organisms carry out thousands and thousands of different endergonic as well as exergonic reactions. And so it comes to this idea of this, compendium, this metabolome, right? So all of the sum total chemical reactions that an organism carries out is called a metabolism. Okay, so I know a lot of us tend to think of a metabolism as being something like just how you process your food energy, or how many calories is in this, or how many calories did I burn doing this run. But it's actually everything. So from growing hair to taking a breath to any of these, like replacing skin cells, that's all part of the metabolism. So it's so much more than that. And so when we talk about things like a metabolic pathway, we're looking at specific functions. So maybe it's just creating new skin cells, or just generating more hair growth, right? So it's not always something that's related to the food you're eating or to the exercise you're doing. And so the other thing about this metabolic pathway is it's a series of steps too, and it's unique usually. Many are very related, very closely related, but each one of the steps in one of these metabolic pathways, and we're going to look at a few of them coming up, such as Krebs cycle, for example. That is a big one, and we'll look at glycolysis and fermentation a little bit, helps to either build or break down a molecule. So it's either going to build a more complex molecule, right? So making more bonds, so storing potential energy, chemical energy, or it's going to break one down. So we're going to release energy, usually like food energy for work in the cell. So endergonic and exergonic reactions in the cell are carried out regularly, but there's a really important molecule that's kind of a segue, literally is a segue between so many of them, right? And that one molecule that's always participating in endergonic and exergonic reactions, transferring energy all over the cell, is known as adenosine triphosphate. So adenosine triphosphate is like kind of small, actually. So it has a little nucleotide like you find in DNA pretty much. It also has a little sugar, and then it has 3 phosphates, okay? So the deal with phosphates is they're really high energy bonds, okay? So they're great for storing and transferring these types, for transferring chemical energy, all right? So we're going to take a little bit closer look at this throughout the semester, but I want you to think about everywhere, right here are 2 of the main arenas where we're going to be storing that energy, right? And well, I guess I'll just show you. All right, so let's take a little bit of a look at ATP and how it works, right? Okay, say we're inside of a cell, and here's maybe an enzyme. An enzyme is a protein that acts like a machine. It usually needs to do some work. Not all, but some enzymes require energy from ATP. They have to be paid in energy in order to perform their functions. Okay, so let's just pretend that this is one of those enzymes here. All right, so let's see. Here's our ATP molecule again, right? So some sugar, some adenosine. All right, so an ATP is fully charged when it has all 3 phosphates. And so say we need to pay this enzyme off to do some work. Our ATP molecule comes up to the enzyme, and it transfers or phosphorylates that enzyme. Basically, this happens. Well, I gotta do some more drawing. Okay, so what happened is this ATP molecule came up to the enzyme that needed to have some work done, and it gave it one of those phosphates. And in the process, it released energy, or it transferred energy to this enzyme. So now this enzyme's like, oh, I can do work now, right? So it's phosphorylated it, and it's left with a byproduct. See how this now only has 2 phosphates? So now it's ADP, right, for diphosphate. So here's our molecule again. This is a little bit nicer version. So here's our adenine, adenosine with the ribose. So there's our sugar. Here's our phosphate groups. And now we've shifted and we've released that energy. So how do we release that energy? Let's talk about that. We do it using a process called hydrolysis, right? And so what happens is we actually add an H2O. Here's a little droplet of water. Not even a droplet, it's literally a molecule of water. And so we've added in a little molecule of water, and by splitting that water, right, hydro meaning water, and lysis meaning to split or to break open, right? We've been able to add it into this molecule and break open the ATP, thus phosphorylating the enzyme, giving it that energy that it needs, and then leaving us with that diphosphate, right? So ADP. So now that you've seen a lot of ways that we can transfer energy, learned a little bit about the thermodynamic laws, and then learned also about this really important molecule ATP, which we will talk about a lot, let me tell you, I just want you to think about all the ways that it's used. So at least in terms of the cell, which is really what we focus on in this class, we're going to be using it for chemical work, mechanical, physical work, and we're also going to be using it a lot for things like transport. So we use ATP a lot in the cell. And ATP is pretty great. Some of the ways that it's really useful is the fact that it's renewable, right? So we can actually recharge it just like a rechargeable battery. So what happens originally is we release that energy, that exergonic reaction, and that's what we looked at mostly in those drawings. What I didn't tell you, and what we'll get to a little bit later when we start going over some of the electron transport chain, is how we can actually recharge it. So we can have, hold on, let me get it going. We used up, we got like an ADP molecule, and then we have what we call inorganic phosphate, and it usually looks like that with a lowercase I. So what we can do is we can recharge it using a handful of different metabolic pathways, and then we're ready to go again. There we go. Let's transfer some energy. So we talked about all these energy transfers. We've really talked about ATP a lot. And I've introduced you to the idea that it's rechargeable molecule. Now I want you to look at this. See what we have here? It's, oops, hold on. This poor pen. It is a cycle, okay? So this represents a pathway, a way we're recharging, reusing, and then using back up this energy that's in the ATP and ADP inorganic phosphate. We've learned about exergonic reactions and we've learned about endergonic reactions. And now we're going to move further along and talk about some of the ways that the cell uses it in very specific pathways. It can get a little intimidating, so feel free to watch these videos over if you need to. And if you need any help or other resources, just reach out. All right, have a great night.